for organizing this nice meeting despite the current situation. So unless we cannot discuss easily together, we have this opportunity to share some recent advances. It's nice, and thank you very much for that. The topic I will present today is in part of a PhD of Elisabeth Barthelemy, which is also connected now to the Zoom meeting.
Hello, so everyone.
Yes, hello. And it is part of the PhD, so we will present the kind of modeling approach we develop in this PhD to test some hypotheses on the influence of past environmental variation on current biodiversity. So it is ongoing work. So what I present is the general spirit of the approach and what we will provide as tools to test hypotheses. And I will briefly explain what kind of application we will do, but it will be only partly more perspective work than results. So I start by the big question that the PhD work will address. So the basic idea is that in biogeography, we are interested in what has shaped biodiversity over time, and even in current biodiversity distribution across continents, we can have the signature of the past history of tectonic, geomorphologic events and environmental change in the past. So if we look at the taxonomic diversity in different places over the world, if we look at the phylogenetic structure, we keep the signature of these past events. But the question is, if we only have a snapshot of extant biodiversity, we cannot see all the details of the dynamics over time. So we have very limited information in the distribution of biodiversity now, at present. And what we want to ask in this PhD work is, can we infer and test the effect of specific past events, which has some long-term influence? And if we can do that, we need to have some proper statistical tools to analyze it from data we have on different ecosystems. So that's a big question we want to address in this PhD work. So for that, we need to combine the evolutionary dynamics and the ecological processes, which both shape and determine the distribution of species now, currently. So one discipline which has emerged and work on this kind of topic is phylogeography. In this case, we use molecular information to assess what is the pace and the nature of past mutation and drift dynamics. So it is basically used within species. So we extend the context of population genetics to integrate variation in the past of the structure of population and habitat variation. And currently, more and more, we want to move from within species perspective, so single species with different genotypes within this species, to a broader perspective of multi-species coalescent in order to investigate how different species could have been shaped by the same broad factors like climate change or tectonic history, which has influenced not only one species, but many species at the same time. So we want to extend this perspective of phylogeography to a multi-species perspective in biogeography. So the modeling approach we need to develop to assess, to ask this question is a modeling approach which is already used a lot in phylogeography, but at the level of within species variation. So here we extend this framework, the coalescent-based approach, to analyze the diversity of species, so between species, not only genetic diversity within species, but also the diversity between species, so the richness, the difference of abundance of species. So we want to extend the coalescent-based approach to a multi-species context. So in a nutshell, we want to investigate what are the plausible genealogies of individuals present now. So that is the individuals you can see at the bottom of the figure. So these individuals, we can see them, count them, and sample them. And depending on the past events of drift, past events of mutation, and past events of migration, we can define some plausible genealogies, which are consistent with scenarios of past environmental change. And then we can determine to which extent the current distribution of individuals, their diversity, is more related to one scenario or to another scenario. So we can try to test if we have some signature in the current composition of community in their genealogy, which reflects this past dynamics. So in coalescent modeling, in fact, we try to find the common ancestor of individuals passed in the time. So that's why we talk about a backward approach. And by doing that, we go back in the past and determine what should be the distribution of ancestors at which speed you can have coalescent events, meaning that you have several individuals at a given generation which share the same ancestors as the previous generation. And the faster it is, the more recent is the common ancestor you can see at the top of the figure. So the shape of the genealogy will depend a lot on this history and the speed of coalescent events. Coalescent events depends on the birth, the death, and the dispersal processes. So we want to represent and simulate this genealogy depending on the probability of birth, death, and dispersal in the past. So here, for instance, we have a scenario where in the past, we can consider that you have some, for instance, a colder period or warmer period with different extent of a given habitat, a given biome, a given ecosystem. And in this case, the effective size of populations should have been smaller in the past than they have been later on in the new environmental conditions. So in phylogeography, we talk about a bottleneck, so which happen at some time, and then you can have some expansion events. And we want to tune, in fact, the extent and the timing of this changing population size and what should be the impact on present diversity. So variant individuals can be introduced in the coalescent using mutation modeling. So in the first basic framework, we just have a kind of point mutation process, which determines that at some generation, a descendant of a previous individual can have a new mutation with some probability. So that's why we talk about a point mutation process. So it's not a mutation in terms of DNA, it's a mutation of terms of, we have a variant which is appearing at the next generation from a given ascendant. So branch can be merged according to different genotypes and defining monophyletic, genetically different and morphological or genetical variation between groups. And these groups can be defined as taxa, a new species. So we use the approach of Monceau to map the genealogy of individuals to a phylogeny of species according to a given speciation process with some specific monophyletic rule and rules also about whether paraphyletic or paraphyletic groups can be merged or not. And according to that, we can transform the genealogy of individual into a phylogeny of species predicted by the coalescent-based approach. So summary statistics, we can simulate a lot of plausible scenarios of coalescent. So we can have a benchmark of communities and different genealogies we have simulated according to parameters of past environmental change. And we can explore the diversity of patterns we have in extant communities for each kind of scenarios, and by tuning the different parameters of past drift, past migration, and past expansion events. So the ongoing work is to prepare and to properly code everything in this framework. So we have a Python library, which is nearly ready. So it should be soon available, and we will provide all the help files, example files needed to easily use it. And over time, we want to refine and provide more tools to have different kinds of scenarios and to use some statistical analysis for testing and inferring parameters with this kind of framework. So we will show what kind of exercise and what kind of application we can have with this framework. So here we imagine we have assess what should be the influence of past restriction, habitat restriction, especially in the case of refugia. So in the past, in many kinds of environment, we have some episodes of habitat restrictions and then subsequent expansion. You can have alternation of this kind of events, but for sake of simplicity, we just started to investigate what should be the signature of a single restriction followed by some time of expansion. So we simulated some genealogies of individuals under this model, and we modulated two basic parameters of past environmental change. A first parameter, which is the amplitude of habitat restriction or expansion after the end of the restriction, and the time at which we have a change of habitat size in the past. So the basic question we want to address is, depending on the amplitude of habitat restriction and depending on the ancient, of the time of past expansion, do we have, and what is the signature of this event on extant community composition? So each color here represents different species. So we start with a coalescent approach and model of individuals in the present composition of community. And then we apply the approach of Monceau to get different lineage with species, different species. So we can define the phylogenetic structure of extant assemblage depending on the scenario. We can also model the evolution of trait values over time to assess what should be the functional diversity of a given assemblage depending on how past environmental change has affected the dynamics. So we can have a lot of simulated composition, and we can assess the trait distribution and the abundance. So what we can find is something which makes sense, is the fact that if we have very old expansion, very old restriction, we lose the signature of this past restriction on extant species richness, meaning that in fact, it's so old that the composition, taxonomic composition went to equilibrium afterwards. And then we have a kind of equilibrium, of mutation drift equilibrium, which determine the extant richness. So we lose at some time the signature of past expansion on taxonomic richness. If we look at the phylogenetic diversity, we have a different story to tell, because phylogeny can show the structure of lineage in the past, and different metrics of phylogenetic diversity can keep the signature of old events. Especially if we look at the bottom one, it is the imbalance between old and recent branching events. And these statistics showing whether phylogeny is more TP or more STEMI can reflect the fact that we have less opportunities for diversification over a period in the past than in the recent past. So I go more quickly because the time is going quick. So for the functional diversity, we can simulate different models of trait evolution. So here, for instance, we have early burst, meaning that trait diversification is more ancient and recently less frequent. A Brownian motion is a regular and over time diversification following a Brownian motion with some variance, which is a sigma here. And late burst is an acceleration of trait diversification in recent time. So what we want to see is to what extent the average, the variance, and the distribution, the shape of the distribution can be affected by the scenarios under these different models of trait evolution. So we can have a kind of baseline patterns that can be used for testing different scenarios once against other. So the idea at the end is to investigate different facets of diversity, taxonomic, phylogenetic, and functional diversity, which together represent different aspects of the influence of past habitat restriction on the composition of assemblage over time. So when we look at these different facets of diversity, we can show that over time, if we have greater ecological drift and higher extinction rates risk in small refugia, we lose lineage, and we can see that in the richness patterns and in the phylogenetic patterns. It depends on how old is expansion after the refugia event. So taxonomic diversity is more related to recent events, while phylogenetic diversity can reflect more ancient effect of these scenarios. And we also expect that according to the speed of trait evolution and the interplay with this changing habitat availability, we should have some signature. So it's kind of virtual experiment showing how we can use the simulation framework to test hypotheses about the linkage between processes and signature. So to conclude on this ongoing work, I will just talk about the last perspective, which will be application to real data. So we will have a kind of large benchmark of simulation using parameters of variation of habitat size, mutation rates, speciation modeling, migration between regions. And we will compare these simulated communities to an observed community you have on the left here. And we will use approximate Bayesian computation and statistics to assess which scenarios and to which extent given scenarios can explain well the observed community. This will be applied to a scenario of diversification we want to investigate concerning the tropical forests which were around the equator over time, but they have changed in a latitudinal extent, and also they have been broken by the drift of the different continents. So they provide a good example of something which has been expanding, contracting, expanding, contracting, and also being broken by events, tectonic events. So we want to see what could be the signature of these past events, which are well known, on the diversity, phylogenetic diversity, taxonomic diversity between regions, and functional diversity currently. So of course, it will be a big work to investigate that properly, but we will use the simulation framework as a theoretical study, theoretical experiment to assess and to test hypotheses on what could have happened in this tropical forest over the world. So I won't go into details, but we will have some hypotheses about the influence of separation on the lineage and phylogenetic diversity between regions, and the influence of migration will affect the beta diversity between different regions. So we expect that, we hope that we will have good signature of these events when we analyze the observed data. So by comparing all these patterns, we hope and we expect that we are able to test some influence of this past history. So to conclude, this as a further challenge we need to investigate. So we want to refine the modeling framework to take into account spatially explicit population distribution. So we can make it depend on the distance for migration, for instance. We want to integrate, in fact, the effect of selection over time on species diversification and traits, and also take into account interactions. So we have a kind of simplistic approach now, and we want to improve it over time in order to do more hypothesis testing and to challenge the different expectations from the different models. So thank you for your attention. So I will be very happy, and Elizabeth also, to answer your questions and to exchange with you afterward.
Okay, so there is a first question from Nicolas. Can you ask your question directly?
Yeah, yes, sure, I can. So very, very nice talk.